Cool. Well, I'm really excited to introduce myself and a new project that's just getting off the ground at Archbold. I know many, but not all of you. Um, I'm Sarah Fitzpatrick, assistant professor based at Kellogg Biological Station at Michigan State University. Being based at a field station is sort of a dream job for me, in part because I grew up at Archbold and really credit Archbold and the people and science that I was exposed to at a young age for pursuing a career in ecology and evolution. So it's fun to get back there now to do my own research. This talk is going to be very data light. My goal is to outline the motivation and plans for this new research and explain what we have going on in these tubs that you've probably walked by if you've been to the station in the last couple of years. And I'm going to start with an iconic story that takes place in Florida that many of you are probably familiar with. That is the recovery of the Florida panther, where in the early 1990s, the Florida panther population got down to around 30 individuals, many of which showed these really extreme signs of inbreeding depression. And it was pretty certain that at this point, Florida panthers were bound for extinction. So at this point, wildlife managers from Florida and Texas teamed up and translocated 8 female panthers from Texas into the state of Florida. And the infusion of this new genetic variation into the small inbred population in Florida led to a reversal of population decline. And this has become one of the sort of iconic examples of something called genetic rescue, which is an increase in population growth due to the immigration of new alleles, new gene flow by more than just the demographic addition of immigrants, more than just the addition of those 8 initial panthers. So there were a handful of early examples of genetic rescue in wild populations throughout the 1990s and early 2000s. So these graphs all show sort of similar patterns of small population sizes that then reverse following the onset of gene flow, indicated by these arrows here. And these early examples were followed by a surge of similar examples from nature throughout the last decade. These all make really compelling stories. They tend to get a lot of attention in the press. But for the most part, these are observational, unreplicated studies kind of representing these one-off type of scenarios. So although our understanding of genetic rescue in the last decade has definitely improved, this is still sort of the unsatisfying state of things with respect to using this management strategy for conservation of biodiversity. The potential is increasingly recognized, but the level of risk and uncertainty are just still too high. So aside from the sort of handful of iconic examples of genetic rescue and conservation, this is not a widely used management strategy at this point. And when it is used, it's typically used in this kind of last resort effort in conservation to avoid extinction. And like I said, this is largely due to these many remaining unknowns and possible risks associated with using this strategy. So how much gene flow is too little? How much is going to be too much and cause outbreeding depression or swamp local adaptation? How do you actually go about doing this type of work? How do you target populations or individuals to use for translocation? How does the magnitude of genetic rescue vary across different taxonomic groups or scenarios? And importantly, can we predict its success? And most fundamentally, what even causes genetic rescue? So the sort of common wisdom is that what causes genetic rescue is primarily works through a recovery of inbreeding depression, but another potential benefit is that this new variation provided by gene flow actually might increase an individual's stress tolerance, or over many generations, over longer timeframes, might increase population's ability to adapt to changing environments. So this was a cover of a recent science magazine that says variation matters, and it refers to this article that showed that individual heterozygosity predicted survival rates in translocated desert tortoises. So a really big focus of my of research in my lab is working to improve the design, implementation, and monitoring of genetic rescue in species of conservation concern, including Florida scrub jays. But because of the unanswered questions and this uncertainty that limits this strategy in conservation, I also use experiments to try to improve understanding of the mechanisms and predictability and outcomes of genetic rescue. So much of my previous work has leveraged the model Trinidadian guppy system to understand fitness and population level effects of gene flow, of manipulating gene flow in wild populations, as well as in the lab. My next slide is just going to summarize over a decade of work on this system into a single slide. So in Trinidad, we studied the effects of new gene flow from translocated guppies sampled from this red downriver locality that were moved into these tiny headwater streams, where there were really small populations of guppies that had low levels of genetic variation. And we saw these dramatic increases in population size following the onset of gene flow, shown by this red arrow here. So these guppy populations started out under 100 individuals each, and by the end of the study were nearly 10 times as large. And then using intensive mark recapture data, genetic assignments, and wild pedigree reconstruction, we were able to show that the increases in population size that we observed were due to hybrid guppies that had longer lives and had more babies. So the gist for these graphs is just that these quadratic relationships were supported, where the intermediate guppies with intermediate hybrid indices had longer lifespans and had more babies. So it was the hybrids that contributed to this really dramatic increase in population size. So this was really strong evidence for genetic rescue in wild populations, and importantly was replicated in at least 2 populations. And then I also got really interested in this idea that variation provided by gene flow might increase stress tolerance. And so I tested thermal tolerance in those same guppy populations that experienced genetic rescue compared to neighboring populations that hadn't had gene flow. And in fact, did find higher thermal tolerance in these populations in blue here compared to guppy populations just in the next stream over that hadn't had that gene flow. So similar environments, but no previous gene flow. And then in a different experiment in Michigan, I also found higher survival rates in populations with previous histories of gene flow when exposed to lethal cold temperatures. So these patterns really intrigue me in the context of rapid global change, where organisms are not only found in increasingly small and fragmented populations, but also facing novel and really stressful environments at times. So what is it going to take from a conservation and management perspective to promote the ability for individuals and populations to withstand first and then eventually adapt to stressful environments as opposed to going extinct? And this is the interest that led to this pilot work that I started at Archbold in 2019, and the NSF proposal that Betsy Rothermel and I recently got funded. In this case, we're using eastern mosquitofish, which are a close relative of guppies. They're found in really high abundances in almost any freshwater body in Florida, including throughout the scrub wetlands. In 2019, I collected fish from one of the cattle ponds out on the reserve and set these tanks up to manipulate evolutionary history in mosquitofish. And so the scenario to envision here is a species with a large contiguous range that then gets fragmented into tiny isolated habitats. And so each tub is one of these tiny fragments where we founded each tank with just 3 fish each to induce a really strong genetic bottleneck. And then you can see this large tank in the back here represents a larger habitat and larger population size that didn't go through this bottleneck. Eventually, half of these tanks are going to receive a small pulse of gene flow to induce genetic rescue. And the eventual goal for this phase is to generate these populations with 3 different evolutionary histories: strong genetic drift, drift followed by gene flow, which is like the genetic rescue treatment, and then one that has stayed large and genetically diverse and outcrossing. So having those populations with these different evolutionary history treatments sets up the ability to address the main goals of this new project. And I'm just briefly going to outline the plan for these first 2 aims. So it's well known that inbreeding depression gets worse in stressful environments. But what's lesser known is the extent to which new gene flow, like in the case of genetic rescue, improves stress response. And so we're going to explore this using thermal tolerance experiments, looking at how fish from the populations that received gene flow differ in how much they can withstand heat stress compared to those fish stuck in the drifted and bottlenecked populations. These are just a few pictures I took last week when I was at Archbold setting up a new fish room in the animal rooms in the back of the RAND building. Big thanks to Betsy and Bert and Loopy for helping to get those rooms ready. They're starting to smell a little bit less like mouse poop. Um, and this is where we're going to be carrying out these thermal tolerance experiments. And then the other big experimental effort that this project will entail is what I'm calling the mega mesocosm experiment. And this is where fish from those 3 different evolutionary history treatments are going to be used to start new mesocosm, 36 new mesocosm populations. Some of those tanks are going to be left at ambient temperatures. Others are going to be heated to near lethal mosquitofish temperatures. And then the plan is to track each one of these populations in really high level of detail using mark recapture methods and genomics, and follow these population trajectories as they either adapt to this extreme heat stress or, on the other hand, go extinct. And importantly, to understand kind of the mechanisms and be able to predict the fate of these populations. So throughout the next year, we're going to be working on building this enclosure behind the Quercus cottage, which is where these mesocosm tanks will go. And once the fish are in there, we're going to be monitoring those tanks for hopefully up to 2 more years, making this a really unique, long-term, multi-generational data set with lots of replication, which is, I think, what the kind of studies that we need to really start improving our understanding of genetic rescue and use it for conservation purposes. So I'm really excited about getting this project off the ground and what we're going to learn from it. I think it's appropriate while set in the highly fragmented Florida scrub to be thinking about the effects of small population sizes and the importance of restoring connectivity and gene flow. And just really want to thank Archbold and especially Betsy Rothermel for supporting and being a collaborator on this project. It certainly would not be possible to do all this without her help. So thank you all for listening.